Now we will see about the compound lipids. Compound lipids are esters of fatty acids and alcohol with additional compounds like phosphoric acid, sugars and protein etc. Compound lipid includes the phospholipids. It is often called as phosphatids, phosphatidic acid and phosphatidylglycerol, lecithin, cephalins, phosphatidylserine, phosphatidylinositol, then lysophospholipids, plasmalogens, and sphingomyelins. We will see one by one. There are other glycolipids. These contain amino alcohol, that is sphingosine or isospingosine. They are attached with amide linkage to fatty acid glycosidically to a carbohydrate moiety, that is sugar, amino acid, or sialic acid. They are cerebrosides or gangliosides. Lipoproteins. They are soluble proteins that combine with transport fat or other lipids, and sulfolipids. We will see one by one. Now, phospholipids. These are esters of fatty acids with glycerol containing esterified phosphoric acid and nitrogen base. They are present in large amount in nerve tissue, brain and kidney, pancreas and heart. Biological functions of phospholipids: they increase the rate of fatty acid oxidation, act as a carriers of inorganic ions across the membranes, help in blood clotting, act as a prosthetic group to certain enzymes. They form structure of membranes, matrix of cell walls, myelin sheath, microsomes, and mitochondria. Now, this phosphatidic acid and phosphatidylglycerol, they are important intermediate in synthesis of triglycerols and phospholipids. You can see the structure of phosphatidic acid here. Second one is lecithin. They are often considered as phosphatidylcholine. They consist of glycerol and fatty acid and phosphoric acid and choline. That's why phosphatidylcholine. They contain unsaturated fatty acids at alpha position and beta position unsaturated fatty acids. Their physical properties: they are waxy white substances and when exposed to air they become brown, soluble in ordinary fat solvents except the acetones. They decomposes when they get heated. They constitute valuable agents for emulsification of fats and oils. Their chemical properties: when boiled with alkalis or mineral acids, choline split off, but phosphatidic acid is also hydrolyzed to give glycerophosphoric acid and fatty acids. They have the property of forming complexes with many different substances like other lipids, protein, carbohydrates, and heavy metals. This property is there concerned with the ionization and functions of many macromolecular cellular components. When the alcoholic hydroxyl group of choline is acetylated, acetylcholine is formed. Their physiological function: in the body, they are combines with proteins to form lipoproteins of plasma and cells. Now these acetylcholines are formed from choline. They have important role in our nerve impulse transmission across the synapses and form the nerve endings to the muscles innervated. You can see here structure of lecithin along with the choline. That's why they are considered as a phosphatidylcholine. Next are cephalin. They always occurs in the tissue in association with lecithins and are very similar in property with lecithin. Only difference is a nitrogenous base. Now phosphatidylserine. Phosphatidylserine, here we can you can see the structure of phosphatidylserine. They are cephalin like phospholipids found in the tissue. Phosphatidylinositol. Okay, here inositol sugar moiety is attached with this molecule. They are found in brain, liver, heart, and soybean. They contain no base but have a inositol in its place. They are acidic than the other phospholipids. Next one is a lysophospholipids. They are phosphoglycerols containing only one acyl radical, and they here you can see the structure of lysolecithin, that is lysolecithin and attached with the choline. Plasmalogen. These are the contents of brain and muscle. These resemble lecithins and cephalin structurally, give the positive reaction when tested with the aldehydes Schiff reagent. They possess an ether linkage at alpha position. Here you can see the alpha carbon, and instead of ester linkage, they possess the ether linkage. The alkyl radical is an unsaturated alcohol. Next one you can see is the sphingomyelin. These are found in large quantities in brain and nervous tissue. Now their concentration of these are increased in Niemann Pick disease in liver and spleen. These contain a complex amino alcohol that is sphingosine, then found in animal tissue, a fatty acid, phosphoric acid and choline. So no glycerol is present. At least three types of sphingomyelins are known in today's time. Here you can see the structure: sphingosine, fatty acid attached to the phosphoric acid, and then again it is attached to the scoline. Together it constitute the sphingomyelin. The next one is glycolipids. Now these contain amino alcohol that is sphingosine or isospingosine. They are cerebroside. They contain galactose, a high molecular fatty acid, and sphingosine. They are also classified as a sphingolipids. They are the chief constituent of myelin sheath, that is cerebrosides. They are differentiated by many fatty acids in the molecule. They are kerosene, cerebron, then nervone, oxynervone. Stearic acid is a major component of fatty acids of rat brain and rat brain cerebrosides. Cerebrosides, especially a cerebronic acid, increases in Gaucher's disease, and the kerosene is characterized by glucose replacing galactose. Now, cerebrosides are much higher in concentration in medulated sheath than in a non medulated nerve fiber. Gangliosides, these are again glycolipids, occur in the brain. They contain sphingoside, fatty acid, glucose, galactose, and acetyl-galactosamine and sialic acid. Now, some gangliosides also contain dihydrospingosine or gangliosine in place of sphingosine. Most of the ganglioside contain a glucose, two molecules of galactose, and N-acetyl-galactosamine up to three molecules of sialic acids. Lipoproteins. They are triacylglycerol, phospholipids, cholesterol, ester, fatty acids, and they combine with the lipoprotein complex. Since pure fat is very dense than water, proportion of lipid to protein in lipoprotein increases, the density increases. Now this property is used to separate lipoproteins in plasma by centrifugation. Density of lipoprotein increases as the protein contents rise and the lipid content fall. Lipoprotein also separated on the basis of their electrophoretic properties and may be identified more accurately by means of immunoelectrophoresis. Now, four major lipoproteins have been identified: chylomicrons, very low density lipoproteins, low density lipoproteins, and high density lipoproteins. Chylomicrons and VLDLs are predominant in triacylglycerol, cholesterol, and concentration of these lipids increased in atherosclerosis. LDL is predominant in cholesterol and phospholipids, increased in coronary thrombosis, atherosclerosis. High density lipoproteins are 27% phospholipids and 45% proteins. The protein moiety of lipoprotein known as apoprotein and constitute 60% of HDL and 1% of chylomicron. Many lipoproteins contain more than one type of apoprotein polypeptide. The larger lipoproteins are consist of lipid core of non-polar triacylglycerol, cholesterol ester, and surrounded by more polar phospholipids, cholesterol and apoproteins. Now, the importance of these lipoproteins are they deliver the lipid to the tissue, maintain the structural integrity of cell surface and subcellular particles, mitochondria and microsome. The beta lipoprotein fraction increases in severe diabetes mellitus, atherosclerosis. Hence, determination of the relative concentration of alpha and beta lipoproteins and pre-beta lipoproteins are of diagnostic importance. Aminolipo- Aminolipids. Now, phosphatidylethanolamine and serine are generally aminolipids, and sphingomyelins and gangliosides contain substituted amino group. The sulfolipids, that is sulfatids. These have been isolated from brain and other animal tissues. Those are sulfates derivatives of galactosyl residues in cerebroside. Now up to this we have seen about the compound lipids. In next we will see about the derived lipids.